Alright guys, today let's look at how CD8 cells are being activated. Now first we have here a couple pieces of clays and we have our antigen-presenting cells. So I'm putting MHCI class 1 receptors on the APC and the antigen being represented with the brown object, and then the B7 coreceptor on the APC cells. Now that I have a blue ball, this one represents the CD8 T cells, which is in this case a naive T cells. So it's going to undergo the first phase of being activated or stimulated. So I use different color of clays to do the receptors on these T cells. Right, so these are the T cell receptors, the alpha chain, beta chain. Okay, and I'm also putting down the CD8 coreceptors. Alright, and then the last bigger piece here is the CD28 coreceptors. Right, so it's kind of bulky in a way, but now I am moving them together to try to make the interactions between the naive T cells and the antigen presenting cells. So the MHC class 1 cells with the peptide antigens there interacted with the T cell receptors, and remember your CD8, 28 and B7 coreceptors are binding together as well. Alright, so I'm taking it apart and trying to mimic the effector T cells, interactions with a target cell, which is virus infected. In a similar fashion, so you have a MHC class 1 presenting the peptide antigens there, okay, in that big piece of red clay. So here I'm making small changes to the T cells, which in terms I'm going to make some of those adhesion molecule. Now, so in simplicity, looking at the interactions, it's very similar. You have your T cell receptors interacting with the peptide antigens, and your CD8 coreceptors are also interacting. Now taking it apart. But before those events can happen, you would need the adhesion molecule. So here what I'm doing with the clay here is to making those adhesion molecule. The one that is on the target cell is the ICAM, and the LFA1 on the effector T cell. Okay, now these are very crude representation, but here shows you the first interactions there, and then remember these adhesions are necessary for the second step, i.e. the actual binding between the T cell receptors to the MHC class 1 with the peptide there, and as well as the CD8 coreceptors. Okay, second contact. Alright, so here is a model with clay showing these interactions between